Okay, thanks for the introduction. My name is Forrest-Hay from Alamar Biosciences. I'm the VP of Sales. Today I'm going to give a brief introduction to our breakthrough immunoassay technology called NULISA, which is a nucleic acid-linked immunosandwich assay technology. And this is really ultra-sensitive liquid biopsy proteomics from Alamar Biosciences. So just to quickly introduce Alamar, we are a fairly new company in this space. So we were founded in 2018 with a mission of powering proteomics or precision proteomics to really enable the earliest detection of disease. And really our mission is to provide the most sensitive proteomics measurements. And we've invented 3 technologies since we were founded that really enables this. NULISA is the technology I'll focus most on today, which is an ultra-sensitive atomolar level sensitivity immunoassay platform that leverages next generation sequencing for the readout and enables protein analysis from either single plex or up to thousands of plex. We've also invented a technology called attobodies. These are biparatopic nanobodies which have picomolar affinity. And this enables us to develop assays for biomarkers where conventional commercial antibodies either aren't available or don't perform optimally. And then thirdly, we've invented a technology or an instrumentation called the Argo instrument, which is a fully automated solution for performing all aspects of the NULISA workflow. So you can go from sample to data very quickly, and therefore enabling high throughput proteomic analysis to be done in your lab. So our mission statement is really to provide automated solutions for ultra-sensitive protein analysis across a range of multiplex levels to detect critical biomarkers from discovery right through to clinical implementation. So at the highest level, NULISA is the most sensitive high-content immunoassay fluid biomarker platform. We have unprecedented sensitivity down at the atomolar level, which is femtogram per mil level of detection. And so that makes it possible now to quantify very low abundant proteins from liquid samples, enabling discovery of critical biomarkers. We can do this across a very wide dynamic range, and we can quantify across a biological range of protein levels within the same assay, making it possible to do high abundant and low abundant measurements from a single sample without any sample dilution. And the chemistry is inherently very flexible, meaning you can go from single biomarker assays right through to high-plex assays with very broad sample compatibility. And with the Argo instrument, you can do this very easily with minimum hands-on time and high reproducibility. So this really sort of sets out the challenge of plasma proteomics, and it's really been one of dynamic range. Most technologies are not able to get down to the very low abundant proteins in the femtogram per mil level. And this is important because a lot of these proteins, especially cytokines, chemokines, and low abundant clinical markers, especially in the neuro field, are present in very low levels, especially at early stages of disease. So by enabling high sensitivity, we're going to be able to find new biomarkers, we're going to enable detection of known biomarkers at lower levels, and also understand what our healthy baseline is when it comes to protein biomarkers. So how do we do this? NULISA is a proprietary technology that we developed, which is based on the proximity ligation assay, but with some very important changes that enable much, much higher sensitivity. And so essentially, we take 2 monoclonal antibodies that target 2 different epitopes on the target of interest. And so this enables very high specificity because both of those antibodies have to bind to create a signal. So to each of these antibodies, we conjugate an oligo sequence, nucleic acid sequence with a known barcode. And so this enables us to actually read the sequence of the oligos as a proxy for the measurement of the protein. And what you'll see here is that on the green antibody, or antibody 1, we have a poly-A tail, or a sequence that ends in a poly-A tail. And on the second antibody, we have a sequence that ends in a biotinylated oligo. And so what this enables us to do is actually capture the immune complex and remove background from the assay to enable much higher sensitivity. And that's really the essence of NULISA, is the removal of background to improve signal to noise and make sure that we can achieve the highest levels of sensitivity. So I'll walk through the assay steps to give you a clear idea of how this works. So the first step of the assay is very similar to any conventional immunoassay. So you basically incubate the antibody pairs into the sample of interest, usually plasma, serum, or CSF. And we use about 10 microliters of sample. So the incubation step takes about 2 hours, and this is the time needed for the antibodies to bind to the protein of interest. And you can see that the oligo sequences end in close proximity to one another. And so that immune complex is formed pretty quickly. But then after that, there's a series of automated steps that effectively remove the background. So the first step is that we introduce oligo DT beads into the sample, and we leverage the oligo DT with the poly-A tail to capture the immune complex. This enables us to essentially perform a wash step, which allows us to remove all free detection antibodies, analytes, and matrix, and it retains the specific analyte bound by the capture antibody. So at this point, we've removed a lot of the background from the sample. We then release that immune complex back into solution and then have a second capture step, which leverages the biotin on the second antibody. And so here we introduce streptavidin beads, which then use that biotin to capture the immune complex for a second time. And then we perform a second wash step on the Argo instrument that removes background and enables us to achieve exceptionally high sensitivity. So at this point, we've basically purified that immune complex. And then the final step of the assay, step 7 of the assay, is the ligation step. So we take an overlapping sequence for the proximal ends of the oligos, and we essentially ligate the sequence together. And then the readout can either be done by qPCR for singleplex assays or next generation sequencing for multiplexed assays. And we leverage typically the Luminex sequencer to perform this readout. So we can actually perform highly multiplexed, highly sensitive immunoassays, leveraging next generation sequencing for the readout. The advantage of this approach is really displayed here. So if you take the same antibody pair and perform a standard proximity ligation assay, which is a homogeneous assay, then you'll see with the red line here that there's background that means that the limit of detection is fairly high, and the dynamic range is not very broad. If you take the same antibody pair and apply the NULISA technology, by removing that background, you essentially lower the limit of detection by about 10,000-fold. So you're now down in the attomolar levels of detection. And you can also see that you create an assay that can measure IL-4 in this case across 7 logs of dynamic range. So not only do we have high sensitivity, we also have a much broader dynamic range for the assays that we develop. Typically, with NULISA, we try and benchmark against the Simoa technology from Quanterix, which has been widely regarded as the most sensitive immunoassay platform on the market. And so here we've developed an assay for HIV P24, again, using the same antibody pair as is used in the Simoa assay. And you can see that we're able to achieve about a 10-fold lower limit of detection using NULISA. And we're down in the 10 attomolar level for limit of detection, enabling us to essentially detect the HIV P24 protein at 0.24 femtogram per mil limits of quantitation. So we've got a very sensitive assay here that is actually more sensitive than the current best-in-class immunoassay technology from Quanterix. And we've now validated several hundred assays, and we've really targeted those assays that are very difficult to detect with conventional ligand binding assays. So here you can see some of the really important cytokines and chemokines that we've developed assays for with NULISA, and we're comparing them here to the similar assays that are available on the Quanterix platform. And what you can see is we can achieve a lower limit of detection, a lower limit of quantification, and that enables us to actually detect these markers in 100% of plasma samples from healthy donor samples. And that's always our mission with the NULISA assay, is to enable detection of all of these low-level immune and inflammation markers and other markers with a very high detectability in healthy patients. Because we believe that if we can measure them in healthy patients, if they're then changing due to some kind of disease or drug response, they'll be able to measure them also in the disease samples. And so just to give some examples of how sensitive the NULISA technology is, here you can see the limits of detection for these 6 different cytokines, and in every case, they're down in the very low femtogram per mil range, which is really unprecedented for immunoassay platforms. And when we do validate any individual assay, here we've got an example with IL-1 beta, which is a very difficult to measure assay. We typically do some very rigorous validation of the assay. including parallelism, spike recovery experiments, and also running it across multiple different sample types. So here you can see we've achieved a higher sensitivity assay than the currently available assay for measuring IL-1 beta with Simoa. We've got a 0.03 picogram per ml limit of detection with very good metrics for parallelism across a wide dilution range, and also spike recovery results that show that we have very little in terms of matrix effects. So we tend to spend a lot of time validating individual assays to make sure that not only is it very sensitive, it performs with 100% detectability and very good reproducibility as well. And here's just another quick example of an NFL, another very important marker in the neurodegenerative disease space, where we've built we've built a best-in-class assay in terms of sensitivity, validated it across plasma, serum, Alzheimer's disease and Parkinson's disease patients, as well as CSF samples. And here we've seen exceptionally good concordance when we compare it to the current assay from Simoa. So again, at the low level, very, very high concordance, and we're able to differentiate between disease and healthy plasma samples. So I think I've demonstrated that we've got the most sensitive immunoassay platform available. But what's really special about NULISA is that not only can you perform singleplex assays, but using the next generation sequencing readout, you can also perform highly multiplexed assays. And so here you can see a set of 5 cytokines from LPS stimulated whole blood samples from 3 healthy donors that were measured both by the singleplex qPCR assay and the multiplexed NGS assay. And the take-home message here is that we can achieve the same sensitivity, the same dynamic range, and high levels of concordance between the 2 different readouts, meaning we can now build panels with highly plexed panels with very high sensitivity. And so that's exactly what we've done. The first panel that we've built is the NULISAseq 250-plex inflammation panel, which provides the most comprehensive coverage of cytokines, chemokines, growth factors, and other important immune markers in a single panel. So you can measure all of these 250 biomarkers from 10 microliters of plasma with exceptionally high levels of sensitivity and detectability. So typically with this panel, we see detectability greater than 98% for all of these markers, meaning that 98% of these markers will be present in greater than 50% of the samples that we measure. And we can measure these across a very broad dynamic range with high levels of reproducibility. What's also nice about NULISA is that we can customize these panels, take subsets of the markers or add on markers of interest with relatively fast turnaround times, and it makes a very flexible platform. So we recently published an online preprint, which is now in review in Nature Communications, which really outlines the technology and goes into detail about this, but also does quite detailed comparisons with both the Quanterix, the MSD, and the Olink samples technologies. So please go to this paper if you want to learn more about the inflammation panel and about how the NULISA assay works. But within this paper, we really demonstrated the power of this the sensitivity by showing that you can detect some of these low abundant cytokines in disease samples versus healthy controls, and really demonstrated the biological power of doing this. So the second panel that we're launching very shortly through our technology access program is our CNS disease panel. And this really leverages the power of the sensitivity and plex levels that we can achieve with NULISA. So here we have all of the key neurodegeneration markers across a wide range of diseases, coupled with all of the inflammatory markers. And so all of these 120 markers in this panel can be measured from very small amounts of plasma, serum, or CSF, really enabling for the first time a very broad view of neurodegenerative disease and neuroinflammatory diseases to be captured from a very small amount of sample with high sensitivity and precision. And just a sneak preview from this paper, from this panel, is that here we ran a small subset of Alzheimer's disease patients, and we saw 33 of the 120 markers in the panel were differentially expressed between age-matched and Alzheimer's disease samples, including some of the usual suspects that are very hot right now in the neurospace. So we can actually measure phosphatide 181, 217, and 231 in the same assay with very small amounts of sample with very high sensitivity. So we're really excited about this panel and the opportunity to now really profile neurodegenerative diseases and understand the real underlying mechanism of disease and differentiate between the different forms of dementia and other diseases. So as I mentioned, we've launched an instrument, and actually Professor Maecker is going to be one of the first labs in the United States to actually get access to the Argo instrument. We've actually spent a lot of time making sure this is as easy as possible to use. So we'll provide the consumables, the instrument, which is capable of running 396-well plates sequentially, and also software to do all of the downstream analysis, including QC and also differential expression analysis to create volcano plots and publication-ready graphics. And so this is really taking immunoassays to the next level. With this instrument, you can have a very simple automated workflow. So it's really less than 30 minutes of hands-on time to set up a run. There's no sample dilutions required. For single-plex assays, you get the data back in 6 hours. There's an onboard qPCR instrument within the Argo instrument. For the multiplexed assays, the output from the Argo is a single sample library ready to be loaded onto the NGS system. So you literally take the sample for that plate and load it onto an Illumina sequencer and run that with a 500 million read run on the Illumina, and you get all of your data back within 8 hours on the sequencer. So this is a high throughput system. You can run up to 396-well plates a day, and you can mix and match between singleplex and multiplexed panels across those 3 plates. And as I said, we provide integrated data analysis and cloud-based analytics with the software and with the instrument. So just one last slide. Right now, we're offering a technology access program, which Professor Maecker actually ran a COVID study with us, which he's going to present shortly. And here you can run our panels or our singleplex assays through our lab in Fremont, California. And there's some details here of how we go about doing that. But if you're interested in running some samples to test out the sensitivity and the coverage of the panels, please contact me, and we can set up a technology access program project. So I'll leave it there. These are my contact details. I'm going to hand over now to Professor Holden Maecker from Stanford University, who runs the Human Immune Monitoring Service at Stanford University, who's one of the first people to generate data using NULISA, and he's going to show some data comparing it to other immunoassay platforms that he has in his laboratory. Thank you very much for listening.
And thank you for that introduction. So it's a pleasure to be here and tell you a little bit about some of the comparisons that we've done in collaboration with Alamar, looking at different multiplex platforms to detect serum cytokines in a cohort of COVID-19 patients. So a few of the things that we're going to go through, I want to spend just a few minutes to talk about methodology of some of these different immunoassay platforms, some of the pros and cons inherent in them, and then get to talking about differences that we saw in detectability between the assay platforms, and as well as the correlations between them. And then finally, a little bit about how we could see differential expression in COVID-19. So most of you are familiar with ELISA. It's sort of the first and simplest immunoassay technology developed over 60 years ago. And of course, there are many types of ELISA. The one that's shown here is called a sandwich ELISA, and this is very common, where you have a capture antibody that picks a target protein out of a mixture sample, and then you have a separate detector antibody that sees a different epitope of that target protein and is coupled to an enzyme like HRP that can then cause a colorimetric reaction. There's by now, of course, a whole spectrum of different immunoassays, many of them multiplexed, that go well beyond what ELISA can do as a singleplex assay. And some of those are sort of shown here according to the number of analytes that they can detect in a single multiplexed assay. So on the far right side, you have the really highly multiplexed assays like the Olink Explore and SomaLogic SomaScan platforms. And over here towards the left side, you have lower plexed assays. The ones that we're comparing that I'm going to talk about today are these 3 in red, which really occupy a kind of middle ground in terms of number of assays that are plexed together, and provide sort of a useful set of analytes that many people are interested in without going to the really, really high and expensive level of plexing of these ones over on the right. So The Luminex, just to introduce for those who aren't aware, is a bead-based assay, and it uses colored dyes in different combinations to create what are called bead addresses that differentiate each of the different kinds of microbeads that are multiplexed together. Each microbead can then become its own little ELISA assay with a capture antibody and a target antibody. The target antibodies in this case are usually biotinylated and then made fluorescent with a streptavidin phycoerythrin detector. And this is all then read out on a modified flow cytometry platform with 3 detectors, 2 to read out the bead addresses and 1 reading out the level of bound detector antibody. Unlike Luminex, Olink is a proximity extension assay that uses nucleic acid tagged antibodies. There's a pair of antibodies that bind to each target of interest in the assay, and these are bound then in solution rather than on beads. And when the correct antibody pair binds, you get an annealing and can do an extension reaction and amplify up the particular oligo of interest for either PCR or sequencing as a readout. The assays that I'm going to talk about today in our comparison are what are called the Target 96 assays from Olink, and these are in fact read out by qPCR using this Fluidigm microfluidic platform that really saves a lot of pipetting steps by allowing you to load all the samples on one side and all of the reaction mixes on the other side, and then it carries out all of the combinations in tiny nanofluidic chambers that really makes it a very efficient assay. And Olink, as I said, these Target 96 assays are feature a number of different panels. The one that we're using, that we used in comparison here, was the inflammation panel. So it has 92 different readouts. And then, of course, there's the Alamar assay, which you just heard all about, so I can skip over this, but just to say that this is a proximity ligation assay, so similar but a bit different from the Olink technology, and of course with this automated Argo readout. And the NULISA assay, in terms of the inflammation panel, is now a 250-plex. The assay, as was used for the comparison I'm talking about today, was closer to 200. But it's obviously the largest of the assays compared to Luminex and Olink that we're comparing here. So just in terms of general principles, there are some advantages to each of these platforms, and I've tried to give what as unbiased as I could of accounting of those. On the Luminex side, I think the major thing that differentiates Luminex in a positive way is that it's flexible to the number of samples you can run, because the kits can essentially be broken up and you can run partial plates. You can also have a big choice of different vendors and different kits that are usually quite easy to customize. Some vendors have nice interfaces, web interfaces to do this, and even to build your own assays. Olink has its own advantages. It has a higher number of analytes than any of the conventional Luminex kits that are on the market, especially with Explore, but even for the target assays. And as I said, you can get this high multiplexing with a very reasonable kind of pipetting setup using the Fluidigm biomark system. It also features the lowest input volume of any assay that I know of, 1 microliter input volume, which is really great for very precious samples. And then, of course, the NULISA assay has even more targets and growing all the time. It has a really distinct advantage in terms of being fully automated, and it also has a low input volume of 10 microliters. Not quite as low as Olink, but definitely less than Luminex. So you might ask, well, Holden, why didn't you talk about absolute quantitation as a feature of these different assays, or not a feature, as the case may be? And that's in part because I don't have a whole lot of faith in that quantitation that's available with these assays, simply because there's error that gets propagated when your standards are diluted over a wide range to like 4,000 or 5,000 fold. And that error means that you are not necessarily quantifying very well at the very low end of the scale. And these curves, these standard curves, are also generally sigmoidal in shape by nature. So at the very high and the very low end, the quantitation is definitely not linear. And that's not just an opinion of mine. There's a paper here in Science Reports from this group that talks about the same issues and comes to the same conclusion. And I can show you also very quickly some data that we generated a long time ago with Luminex showing this exact phenomenon, where we ran the same sample 3 times and measure a readout of here just showing you like 9 cytokines. Either the MFI, the median fluorescence intensity, which is the raw readout, comparing the 3 runs, or converting that to concentration. And I think you can see that the 3 runs of MFI are very tightly clustered, whereas for at least some of these analytes, the concentrations seem to differ quite a bit more. So with that, I can start talking about our comparison of these 3 assay platforms in terms of COVID-19 samples that we had and some healthy controls that we included as well. Again, we'll talk about correlations, detectability, and ability to predict severity, as well as changes over time in the COVID-19 samples. So these samples came from a very large study that was NIH funded that we participated in as a core lab called IMPACT. It was a study of hospitalized COVID-19 patients starting early in the pandemic. And there's a couple publications here that describe the study design and some of the clinical findings from it published in the last couple of years. It was a very intensive sampling study with lots of visits early during the hospitalization period and then also during the convalescent follow-up, and lots of different multi-omic assays, including Olink, as part of the study. And then we used a small subset of the samples that we ran on Olink to also run the other assays. So we were one of the core labs doing, as I said, Olink, but also CyTOF for this particular study. And there was a central data analysis group that put all of these data together and has a portal, and these will eventually all be made public. And this is now the latest paper from the IMPACT group down here that shows some of the multi-omic analysis that was done and some of the biomarkers that have come out of this. And just to show you the small subsample that we have so far run with our comparison study from this large study, it's 25 samples with multiple time points for many of those samples. You can see here the range of different visits that were included, and these trajectory groups, which indicate the severity of the outcome from mildest being number 1 to fatal being number 5. You can see that most of these are actually relatively severe patients, but there is a spread of trajectory groups in these in this subsample. And so the first thing that you can ask is, can we differentiate the COVID-19 patients from the healthy controls that we included in the same assay? And I just show this slide to start because I do want to make the point that with more targets, as we have in the NULISA assay, you, of course, can find more differentially expressed proteins. So with 203 targets, there's lots of things up here that come out as differentially expressed by NULISA, somewhat fewer by Olink and by Luminex, because in part, they have fewer targets to begin with. I don't know how to explain the fact that Olink is picking up more lower expressed proteins in COVID-19. I don't know if that's an artifact or something to do with the targets that are included in the Olink assay. But anyway, the main point being that more targets gives you more chance to detect differences. So we thought, well, let's kind of home in on those targets that are common between at least pairs of platforms. And so here initially I'll show you some data with pairs of, in this case, NULISA and Olink. And there are 56 targets in common between those two platforms. And you can see that, yes, NULISA, even with just those 56 targets, is still detecting more upregulated genes in the COVID-19 than is Olink. But again, Olink has a few more of the downregulated genes that are popping up in that platform. If we turn instead to the targets that are common between all three platforms, that turns out to be just 33. And you can see that still the Alamar assay has more upregulated targets. Olink, again, a few more downregulated, and Luminex is kind of in between the two in terms of having not quite as many upregulated as Alamar, but a little bit more or similar, I guess, to Olink. And so here is the summary of those data. You can see that all three platforms identify a number of differentially expressed proteins, and those are the ones in the middle here. And then there are subsets that are only partially overlapping, as well as some that are uniquely found by one platform only. Again, the Olink has a number of them, some of them downregulated and not upregulated. And what I've drawn in gray here are analytes that were differentially expressed in Olink, but that also had relatively poor detectability in the assay. So that may be somewhat of an artifact in terms of their differential expression. So that's differential expression. Let's move on and talk about correlations between the assays. Definitely, there are some correlations. And what you can see here is a plotting of the Kendall coefficient of correlation between pairs of these assays. And so for Alamar NULISA versus Olink, there's actually a pretty good correlation in terms of a number of analytes that have a high coefficient for correlation. When you look at NULISA versus Luminex, it's a little bit more spread, and there's a pretty good subset down here that are actually poorly correlated, similar to Olink. And in fact, Olink may be even a little bit more dramatic when compared to Luminex, that there are just a subset that really don't seem to correlate very well between those two assays. Perhaps not so surprising, because they are in fact very different technologies, whereas Alamar and NULISA are at least a bit more similar in terms of the type of assay and the readout. So you see here in the bottom these coefficients plotted, and you can see that the best correlation is again between NULISA and Olink. Here's a very, I think, telling slide looking now at the sensitivity of these assays relative to their limit of detection. And again, we're limiting in all of the slides hereafter to just the 33 common targets between these platforms. And what you see is the range of values for the COVID-19 samples for each of these shared targets, and the red line indicates the limit of detection. So in the Alamar NULISA assay, there are really only a couple of targets for which many of the samples are at or below the LOD. In Olink, there's actually a whole set here of targets that are right around the LOD. Similar in Luminex, although it's a little bit more of a continuum, whereas in Olink, there's kind of a break, and then there's these really well-detected analytes coming up above. So before we cast too much shade on the Luminex side, I want to also mention that there is a bit of an improvement with the Luminex assay if we do a correction for non-specific binding. And this is something that's possible using these so-called assay checks beads, the checks 4 beads that are part of that spike in control set of beads is a non-specific binding bead. So it measures background or non-specific binding in the sample for each of the channels. And by removing that background, we actually improve the detectability a little bit for some of these ones that were really dropping below the LOD over here. They're still close, but they're slightly improved by the removal of that non-specific background. And that's something that's actually unique to the Luminex platform that we don't have an option for in terms of a quantitative correction on the other platforms. And so again, showing you that in a different way here, with and without the correction, you can see that the Luminex detectability is improved over here after the non-specific binding correction, although some of these are still a bit lower in detectability than they are in the other two assays. And again, the Olink has this set of half a dozen or so analytes that are just poorly detected in that platform specifically, where the Alamar platform has only a couple that are falling down there. So now to talk a little bit about predicting the outcomes or the trajectory groups for the COVID samples. I want to just introduce this idea of clinical trajectory groups that was kind of developed in the impact study. And here, this shows you the score in terms of clinical ordinal scoring that's done on each day post-admission for the patients in the study. And you can see these different trajectory groups can fall from the most mild, which rather quickly recover, to a score of 1, which is the mildest. And then you have the group 2, which just takes slightly longer, but also comes down to a baseline of 1. The rather slower yet, but still never too severe case is trajectory group 3 in blue. And then you have group 4, which is severe for a longer time, group 5, which becomes fatal. So with those groups in mind, we had such a small subset of samples in our sub-study here for this comparison that we decided to group the trajectory groups 1 through 3 together, calling them mild, and 4 and 5, calling them severe, and then just use that as a outcome to see which of these assays could differentiate those groups the best. So I'm going to focus here on just the comparison of NULISA and Olink, since those were the most correlated assays. And you can see here that both of them actually managed to pick up at least a few analytes that are differentiating in these volcano plots, the severe being upregulated versus the milder trajectory groups. And you can see also their longitudinal plots over here on the right side over time. And you can see that they maintain their differentiation pretty much, the red lines from the blue lines being the severe versus the less, more mild cases. And so then what about using a kind of way to dimensionality reduce the data and look for separation of these different trajectory groups? This is something called a star plot, which was invented by our senior statistician Tyson Holmes as a way to, using pre-selected outcomes, find that plane through the multidimensional data that best separates those outcome groups. And so here each number is a sample, and the color and the number encoding is the trajectory group. And so with the Alamar assay, you can see that the trajectory groups can be fairly well separated, with 1s and 2s out here, and 3s and 4s and 5s. And you can also see which vectors here in terms of the analytes are most differentiating for those particular outcome groups. Olink also does a reasonable job with somewhat different vectors differentiating the groups, but you can see again 1s and 2s, 3s, 4s, and 5s being kind of separated in a counterclockwise manner. So again, just another way to look at the ability to separate these groups, and both assays are able to do it more or less well. What about changes over time? I want to just show that, again, NULISA and Olink can both differentiate those analytes that are changing significantly over time. And you can see there's a number of them. There are a few more, though, in the NULISA platform than in the Olink, again, focusing just on the common targets. And another way to look at that is here with this two-dimensional plot, where you can see the ones that are significantly differentiating in NULISA versus in Olink over here by chi-squared statistic. And indeed, there are more on the NULISA side that are reaching significance compared to Olink. So more things being picked up as significant changes over time. And that's summarized here again in a Venn diagram. There are a couple of things that are uniquely picked up by Olink, one being the CCL3, which you may recall was on the not so great list for NULISA and was one of the two that were falling lower in detectability. And I'm told that the company is working on new detector antibodies for that particular target. So that was a not surprising find for Olink to be doing better. But there's a whole set over here that are differentially detected by NULISA and not by Olink, including all these ones in blue, for which there is in fact higher detectability in NULISA versus Olink from the comparison I showed you earlier. So to conclude then, I just want to show that we've been able to find correlations between NULISA and Olink assays that are reasonably good, a little bit less well with Luminex, but still some analytes that are actually well correlated between all three. We found that there are more samples below the limit of detection for Olink and Luminex than there are for NULISA when we look at the range that each of these assays has for the COVID-19 samples that we used. All three assays were actually able to predict COVID-19 clinical trajectory groups fairly well, and I should have mentioned that that was always from the baseline admission sample, so that's pretty interesting. The NULISA assay showed a little bit better ability to find longitudinal changes over time in the same impact patients. And of course, NULISA has the ability to multiplex more targets, in this case over 200, with excellent sensitivity. And so that allows it to find even more things based on the number of targets that are available. And so with that, I want to acknowledge folks that contributed in our lab to these data: Tran Nguyen and Koji Abe. Yael Rosenberg-Hassan ran some of the initial Olink assays, and Tyson Holmes, our statistician, has helped with some of the analyses. On the Alamar side, big thanks to Joanne Beer, who contributed a lot of the plots that I showed, and Alex, who has supported us from the beginning and ran these samples on the Alamar platform in their facility. In the Impact Network, of course, there's a whole group of people and sites that contributed the samples we looked at. Some of the leadership is listed here, so a big thanks to them for allowing us to do this study, and we intend to actually continue on the NULISA platform with analyzing the rest of the impact samples so that we can increase the breadth of data that we have for serum proteomics in the study. So with that, I'd like to thank you for your attention and turn it over for questions.